present not only to the FDA and to the Institutional Review Board at the University of Florida, but also to this Data Safety Monitoring Board, which is comprised in our case of both gene therapists and non-gene therapists, but also neuromuscular clinicians. And in unanimous agreement with all three groups, we dose escalated to our higher dose and have been moving forward very quickly to generate data at that higher dose. And to take a step back, and again, because I have the privilege of talking first today, the doses I mentioned that we moved into is this 2 × 10^14 vector genome per kilogram dose, which is similar to the dose range that our colleagues at Sarepta are working in, as well as our colleagues at Pfizer. And so we're all revolving. I think it's very exciting, actually, that we're all moving diligently and very quickly to dose and understand how these gene therapies work. And then importantly, there seems to be consensus about the dose range that we all need to be in to achieve robust efficacy, also balanced against safety. So the last thing I'll talk about is just about our manufacturing strategy, which is a key cornerstone of our company. But I would say that it's no surprise that before we dosed our first animals, we began working critically on how to manufacture these viruses. And I can honestly tell you today that nobody in this field has a definitive answer for how to manufacture enough of this virus to supply the entire need of this massive population. Most gene therapies early on, historically, were being produced and provided for by small clinical centers, academic centers producing gene therapies. And now that there's so much corporate and biotech interest in these gene therapies, we've all begun working very systematically on methodologies to scale these manufacturing approaches. But we're still all very in the early days. And I can tell you that I personally don't believe there's any one single methodology that is the unique answer to solve manufacturing across all these different diseases. Today, I'm sharing with you one methodology for manufacturing AAV. Sarepta is using another, and Pfizer is using a different one. And importantly, we're all demonstrating and showing success in our own independent methodologies, and I think that's very exciting. I think that speaks to how rigorous we're all working and how important it is to work with technologies that you have experience with and know how to move forward. And so nonetheless, we began scaling this manufacturing methodology almost 5 years ago at this point, and it started with a very small-scale process that came out of the University of Florida, which, as I mentioned before, is actually where we're carrying out our clinical trial. Today, our manufacturing process is being carried out in bioreactors at what we consider a fairly scaled point, at a 250-liter scale. And that scale allows us to provide for multiple patients from a single bioreactor. And in the future, we're going to continue to scale this manufacturing methodology to larger volumes. And I think what's unique about our manufacturing methodology, although as I mentioned before, there is no single answer to do this, one of the strengths of our manufacturing methodology is that it uses bioreactors, which are commonly used for other types of biologics. Monoclonal antibodies are also produced in bioreactors. And so we hope, and one of the reasons we chose this methodology is because it uses already established manufacturing approaches. But as I mentioned before, over time, we're going to continue to scale this and move forward with larger scaling up, so increasing the volume, and also scaling out, putting multiple bioreactors side by side to supply as much vector as we can. And so with that, I'll close, and thank you very much for having me again today. It's been a pleasure to share with you our progress so far, and I look forward to future conversations today and from hearing from our colleagues about their important work as well. Thank you.